It's Fátima Sanchís Calleja, everybody!
Guten Abend, meine Damen und Herren. And welcome to B.Y.O.B, or Build Your Own Brain. Tonight, what I want to do is show you a little bit of what scientists are doing in the lab to reproduce a tiny brain on a dish. And of course, these are not real brains as we know them, but instead tiny 3D brain-like structures that we call brain organoids. And we use them in the lab to model human development of the brain, and also to investigate some disorders that are associated to it. So for now, I think we're quite far from modeling the adult brain complexity, but this field is really rapidly evolving, and you will see, hopefully tonight, I'll show you how exciting this research is. Let me present you our cell culture hood. This is an extremely sterile environment where we will work tonight. And it has to stay like that, so please be very clean. Let's get started. What do we need to make a brain? What are we made of? Please.
Water.
Thank you. I didn't expect less. We don't need any kind of cells, but we need stem cells, which are the only ones that are pluripotent, which means that they can make any type of cell in the body. But life is not a bed of roses, and sometimes these cells differentiate, which means that they become a certain cell type of their choice, just like that, and we cannot make any more organoids with it, so we really don't want to see that. So, if we have our cells pluripotent, we can just tickle them with a little bit of chemicals and get them in a single cell suspension. And then they will be jamming and chilling and talking to each other and getting closer and closer until they get stuck and form what we call an embryoid body, which is a tiny pluripotent structure that we can turn into anything, any tissue that we want. What do we do with this embryoid body? So we need to make something more specific with it. We need to make the progenitors of the tissue that we want to build. So I prepared some media tonight. So I prepared this media, but I forgot to label it. So I need your help to decide which media to take. I know that the ones on the right, I think they're used later on in the protocol, but I'm doubting between the orange and the magenta, depending on the color that you see. So I need to see hands raised for the orange. Orange, okay, and magenta? Okay, great. Let's choose the magenta. So we're gonna go to magenta. Can I get a bit of drumming? Awesome. You were right, congratulations, please. So I can tell you that these work because you see this aura here of more translucent tissue. Those are the neural progenitors that are actually proliferating. So I have to tell you that if this was the neural induction media that's making these neural progenitors, I think the other one was probably for intestinal organoids. I think it was Umut's, my colleague, he's here too, so he must have forgotten it there. So, we have our neural progenitors, and what do we need now? We need to make neurons with them. And I'm going to give you 4 options. What can we do with it to get some neurons? We can either attach these bodies to a surface, maybe that helps them grow. We can also embed them in the scaffold to give them some support. We can also change to a different media, or we can split the organoids in 2 parts. So, hands raised for A. Good. Hands raised for B. Nice. Hands raised for C. And finally D. Okay, quite some people. I have to tell you that B and C were right. So, let's see what actually happens when we put some neural differentiation media and we put a droplet of matrigel, which is the matrix that acts as a scaffold for these tiny brains. What do you think? For me, this is amazing. Like, does it remind you of anything? I mean, yeah. So we have these huge ventricles forming, like in more or less like in the human brain, but still the neurons are not quite there. So they are a bit immature, and we need to make them more similar to the ones that we actually have. And for that, we need to put some maturation factors in our differentiation media. And hopefully in the end, we will get a more mature organ. This one has like, these are the arms of the neurons coming out happily. And I'm just very curious, how many of you are reading this singing? Okay, nice, you had a childhood. And a spoonful of factors helps the tiny brains grow up. The tiny brains grow up. Oh, tiny brains. Who wants to have a look inside? All right, this is the organoid. So, okay, this side is quite disappointing, but I can tell you that that side is really awesome. Why? Because this is a human brain in development at 10 weeks. This ventricle here is this ventricle here are the progenitors. The green is labeling the progenitors. And the magenta is labeling the neurons, which in the human brain are coming out of the ventricle and going up. And this is really amazing that we can do this. So let's have a look at the other organoids. What do you think about them? How do they look like? I think we have several problems here. They look quite different from each other, right? Also, their inside is dead. Someone can relate. And then I can also tell you that we have some missing cell types that we have in our brains, but they are not in the organoids. So what can we do with this model? Like, is it actually good to use it? Let's take a step back. So here we have our organoid and our human brain in development. The human brain looks quite complex in comparison, but well, the other option that we have to investigate these diseases and our neurological traits is a mouse brain, which looks really rudimentary in comparison. And we also know that the mouse cells do not really behave like the human cells. So I think the human organoids really deserve a chance to go back and try to improve them. And this is what I'm here for. In my PhD, what I tried to do is make these brain organoids a little bit more reproducible and stereotyped, so that we have an animal-free model to study the unique traits of the human brain during development. And with this, I want to thank you all, and I hope you have enjoyed Build Your Own Brain.